Okay, hello guys. So welcome back to返學 DSE for DSE Biology Chapter 5 of revision videos. Okay, in this chapter we're talking about food. For food, let's have a look at why do we need food. There are several reasons. So energy how is why we need food because it provides energy for our daily activities and also keep us warm. And also food will provide some more materials for our growth and repair. And in biology, growth and repair are two separate concepts. So in your exam, remember to write both terms down, okay? And also food will maintain our health. So generally, there will be 7 types of food substance. The first three, there's the carbohydrates, lipids, and also proteins. They all provide energy, while for minerals, vitamins, dietary fibers, and waters, they do not provide energy, but they are critical for your health. And for carbohydrates, it is made up of carbon, hydrogen, oxygen, and hydrogen and oxygen are the constitution for water. That's why in Chinese we call it 碳水化合物. And for carbohydrates, we classify into 3 groups: monosaccharide, disaccharide, and polysaccharide. And their basic monomer will be saccharides. So it depends on how many saccharides do they have. They are different types. And they are bonded together with the covalent bonds. In order to know more about covalent bonds, I suggest you take DSE chemistry or you have a look in the for IAL series. And in order to make the covalent bond, we need condensation, that is the removal of water. And in order to break it, we need to do the hydrolysis, that is the addition of water. And for monosaccharide, it's the simplest form, and the best example will be glucose. It is sweet and also soluble in water, and they are commonly found in fruits. For fruits, they have glucose or fructose, and for milk, it is galactose. For disaccharide, it consists of 2 monosaccharides, and the examples are maltose and sucrose, and they are both sweet and soluble in water. And for maltose, it is found in berry, and it is made up of 2 glucose molecules. And for sugarcane, it is where the sucrose will be found. And remember, for sucrose, it is made up of glucose and fructose. Okay, the constitution is important because it is a common critical learning point for most students in DSE. And for most monosaccharide and disaccharide, they are called reducing sugar because they can form orange precipitate when they are tested with Benedict solution. However, this exception is very important. Sucrose is not a reducing sugar because it has a negative result in Benedict solution. And for the term reducing is actually linked to the chemistry concept, which is called the redox. In order to know more about redox, I advise you to take DSE chemistry or have a look in the 放學 IAL series. And for the polysaccharide, it consists of many monosaccharides. And example will be starch. They are not sweet and they are insoluble in water. Actually, there are other than starch, glycogen and also cellulose are also common examples in our curriculum. And you have to remember each of them, what are their function and where can they be found. And for the function of carbohydrates, it is served as the main energy source for the body. Remember, the term main is extremely important because other than carbohydrates, lipids and proteins can all provide energy, but the main energy source will be carbohydrates, and it can serve as a energy reserve in form of glycogen or starch, depends on which organism you are. And it also serves as a source of the dietary fibers, which is easy for digestion, that is for living the feces. And in order to test for carbohydrates, there are different tests. The first one is the glucose test paper to test for presence of glucose, and the exact color change is no longer important because you just say color change is fine. And in order to test for reducing sugar, we use Benedict solutions. Boiling is required in this test, and then you may find brick red or orange precipitate afterwards because the copper 2 plus ion, which is blue in color, is now reduced to copper 1 plus, which is orange or brick red in color. And in order to test for starch, we need iodine solution and we do the iodine test. And the color change will be brown to dark blue. So this one is brown and this one is dark blue. Or someone may also say it's a blue black. It depends on you, but both are fine. And for the lipid, it consists of carbon, hydrogen, and oxygen. It is insoluble in water but dissolved in organic solvent. So organic solvents can either be like alcohol or thinner. An example will be triglyceride. It is made up of one glycerol and three fatty acid molecules. And again, they are linked up by condensation. In order to break it, you need hydrolysis. So here is a drawing of a common triglyceride. And there will be at least two types. That is, it is solid in the room temperature form, so we call it fats. And if it is liquid in the room temperature form, we call it as oil. And they are commonly found in milk, meat, or nuts. And for the function of lipids, it serves as an energy reserve, as a shock absorber to protect our internal organs, and also it acts as a heat insulator. And it also helps the absorption of vitamin A and D, which is called as lipid-soluble vitamins. And we also need lipid to produce some hormone, but not all. And also fats, that is the lipid, is also an important component on your cell membrane, which is already mentioned in your chapter 2 and 3. And in order to test for lipid, we do the grid spot test. We have a translucent pot on the filter paper, which is then can be removed by the organic solvent. In the previous curriculum, we will say that as a permanent translucent spots. Okay, so it depends on which year are you referring to. So I prefer the permanent translucent spot, but now we prefer the test to do it in this way. And for the protein, it is made up of carbon, hydrogen, oxygen, nitrogen. Remember, all the protein will definitely have nitrogen. And whether it is sulfur or not, it depends on the amino acid. And that's why the monomer of the protein is called the amino acid, or we call it peptides. And they are also linked up by covalent bonds, but this time the covalent bond is special type. We call it as the peptide bond. Again, it is through condensation and hydrolysis. You can break it down or form it. And then for the structures, we will first determine by the basic sequence, and then the protein will fold to a specific shape, and it may combine with other proteins, which is similar to your concept in enzyme topic. And the sequence is actually determining the final shape of the protein, and each shape is called unique. Unique is also known as specialized or specific, and because of the following examples: enzyme, receptors, and antibodies. For receptor, we will talk about it later on. And for antibodies, we will also talk about it in our body defense topic. And if we change the shape of the active site, or we change the shape by increasing the temperature, it may result in a loss of function, which is also known as denaturing. And there will be two types of amino acids. 12 of them is called the non-essential amino acids, which means that our body can produce our own. However, there are also essential amino acids, which means that we have to obtain it from our diets. And the common food source will be fish, meat, eggs, and beans. And here are some functions for proteins. Again, it is for growth and repair, and it is producing enzyme, antibodies, hemoglobins, and also hormones. And also, if we are really desperate in starvation, and it may use to provide energy. And if we don't have enough proteins, we have this disease, and this disease is extremely difficult to pronounce, so I'm not going to mess with it. But remember, the symptom is that the patient will have a swollen abdomen because of the accumulation of body fluid. And in order to test for protein, we use protein test paper. Again, the color change is not important. You just say color change is fine. And for the fate of protein, remember, at the end of the day, it will be broken down into amino acid and used for different functions. However, it cannot be stored directly. That's why it will undergo a deamination in liver and excreted in urine as urea. This part is important because it is a critical function for your liver. And for minerals, we will include three of them. That is calcium, iron, and also iodine. Each of them will have this format: source, function, and also deficiency. So remember, how do we take in? Like calcium as an example, milk and tofu. Function, to make our bones and teeth strong. That's why the deficiency is the weakest. That's the weak bone. And for iron, the source is liver and beef, and the function is to make hemoglobins. If we don't have enough hemoglobin, we will suffer from anemia. And for iodine, the main source will be seafood, and the function is to providing hormones in producing hormone in the thyroid gland. And if we don't have enough iodine, we have a gouty. And for vitamins, we have three types in our curriculum. That is A, C, and D. Remember, only vitamin C is water soluble, while the other two are lipid soluble. For vitamin A, the source is milk or fish liver oil, and the function is to form a pigment in the rock cell. And because rock cell is required in the night vision, that's why if we don't have enough vitamin A, we will suffer from night blindness. And also in the carrot, we have the carotene, and the liver will convert the carotene into vitamin A. That's why it is not technically correct to say that the carrot contains vitamin A, because it actually contains carotene, and then carotene is later on converted. For vitamin C, the source will be lemon or orange, and I expect students to write concrete example instead of just fruits, which is really embarrassing in many cases. And vitamin C is required in healing wounds and also our immune system to work properly. And if we don't have enough vitamin C, we will suffer from scurvy, and scurvy is closely related to pirate behaviors. And I suggest you to ask your teachers for more information. And for vitamin C, we have a food test called the DSE test. This blue solution, blue DSE will be decolorized if vitamin C is present. And for vitamin D, the source will be fish liver oil, and also if we're exposed to sunlight, our body can also make vitamin D. And the function is to make our bones and teeth strong because it helps the absorption of calcium and phosphate. That's why if we don't have enough vitamin D, we will also suffer from rickets. And for dietary fibers, it is actually the cellulose. And because our body cannot digest cellulose, that's why it will then pass through our body directly. However, during the process, it will stimulate peristalsis. That's why it helps the feces to pass out of the body easily. Don't use the term start with S and with T in your exam, which is extremely improper. So the term is called feces. And if you have a very hard feces, the technical term is called constipation. Thank you. And here are some functions of water. Just do your best to remember 2 or 3 of them. It is a reactant for many chemical reaction, and it is a medium for a chemical reaction, and is a medium for transport, and it also serves as a cooling agent to regulate your body temperature, and is a major component for many lubricating fluid. And for the concept of balanced diet, it includes age, sex, level of activities, and also body condition. So remember, each factor will affect it. And for the term body condition, is actually linked to this term, pregnancy. So different person, because of their activities level, they may need different fruits. Also depends on your situation, you may make your own judgment and comment. So here are the extract from the HKEA document. So you may notice that for this part, it is also closely related to chapter 6, which is then related to the digestion and also absorption, because these 2 chapters are closely related. And in these 2 chapters, there are a number of practicals and experiments. And I strongly suggest you to read those experiments carefully, because with amendment, they may appear in your exam also. So you may notice there are also including practical work. So here will be the end of this video. So see you guys next time. Bye.